Microbial contamination in cell culture alters the outcome of experiments and the growth of the cell culture. To mitigate this, antibiotics are commonly incorporated into the culture medium. Antibiotics either destabilize the bacterial membrane, inhibit cell wall formation, influence the protein biosynthesis, or influence DNA replication and RNA transcription. While these mechanisms effectively kill or hinder bacterial growth, they can also affect the metabolism of the cultured cells. Furthermore, due to the risk of antibiotic resistances, their usage should be avoided. We tried to look for an alternative approach to replace antibiotics in cell culture. Traditional medicine has long relied on various plants for treating diseases and infections. Some of these plants contain herbal agents that possess antibacterial properties, making them potentially suitable for use in cell culture to prevent contamination. During our research for this project, we discovered 3 promising compounds with antimicrobial and antifungal properties. The molecules cineol, thymol, and benzyl isothiocyanate demonstrated a broad spectrum of activity against both gram-positive and gram-negative bacteria. However, due to the supply issues with cineol, we were only able to test the other 2 compounds in our experiments. Our main experiments were an inhibition test to see if our agents are antimicrobial, a serodilution to determine the minimal inhibition concentration, and a resasurin assay to evaluate potential toxicity of the tested compounds on our cells. We started our project by making agar and broths for the tested microorganisms, E. coli, Lactococcus lactis, and Saccharomyces cerevisiae, also known as yeast. Each microorganism had a specific medium to ensure optimal growth conditions. We used TSA for E. coli, Saboro for yeast, and M53 for Lactococcus lactis. Afterwards, overnight cultures were prepared to lay the groundwork for further experimentation. We performed serodilutions of thymol and benzyl isothiocyanate along with the microorganisms to determine the respective minimum inhibition concentration of our agents. As you can see, promising results were obtained for thymol, with E. coli at 0.2 mg per mol, Lactococcus lactis at 0.3 mg per mol, and once again with Saccharomyces cerevisiae at 0.2 mg per mol. However, due to its lack of inhibition on microbial growth and hydrolysis in water, benzyl isothiocyanate was excluded from further experiments. We used the cell line MDA-MB231 as the experimental model in our study. Given that we did not incorporate antibiotics in our cell culture, it was crucial for us to prioritize the maintenance of sterility within the cell culture medium in our experimental setup. To achieve this, we implemented a sterile filtration process once fetal bovine serum was added to the medium. This step helped to maintain a contaminant-free environment for our cell culture. Prior to conducting any experiments, the cells were cultured for a duration of 2 weeks. This allowed them to reach an optimal state for the following procedures. During this period, the cells were carefully monitored to ensure their growth and viability. The cells were passaged twice a week to promote their continued proliferation and prevent overgrowth. In order to assess cell viability, we employed the resasurin assay. In the first phase, cells proceeded at various concentrations. By measuring the absorption at 600 nanometer and 507 nanometer, we aimed to determine the optimal number of cells required for accurate viability measurements. In the next step, cells proceeded at the determined optimal cell concentration, which in our case was 70,000 cells per milliliter. After 2 days of culture, we included thymol in our cell culture medium. Unfortunately, the addition of thymol led to cell death, which ended our experimental phase. Our aim was to explore the possibility of replacing antibiotics in cell culture with natural substances. We hypothesized that these compounds may utilize different pathways compared to traditional antibiotics. However, further experiments are required to investigate this potential alternative. One aspect that needs to be explored is the duration during which the cells are exposed to the substances. By testing the substitute at different time intervals during cell culture, we can gain insights into its potential benefits and limitations. Another crucial factor to consider is the assessment of the substitutes with different cell lines. Different cell lines may show different sensitivities to the substances, and understanding this is vital to determine their potential usefulness. Overall, the outlook of our research involves conducting further experiments to determine the effectiveness, safety, and suitability of natural compounds as substitutes for antibiotics in cell culture.